Well, we are going to start the first moment of EPIA 2013. As you know, this is the day for the challenge initiative, that is a parallel initiative with the EPIA. We thank you all being here, and welcome to Angra, welcome to Açores. We start this journey with the challenges, and this is, as I said, a parallel initiative to the 16th Portuguese Conference on Artificial Intelligence. The challenges have been organized by myself and Pedro Cardoso, and then also by Luís Gomes, and two distinct moments emerged. The first one is this one, which will take all the morning, and the other one will be after lunch, that will be present later on. What was the initial idea of this initiative, the challenges? Well, its main goal is to promote this discussion and ultimately lead to new projects, new ideas, to create links, connections among researchers from different areas of research and artificial intelligence. This is not because AI is a discipline disconnected from other fields of research. We all know by its own nature, AI is a multidisciplinary field. The point is to find opportunities to AI expertise to solve problems in areas where AI is not being considered as a solution. And the reverse also, to show how artificial intelligence has been tackling different areas and how solutions presented by AI can be replicated in another areas, contexts, in a way that was never thought before. That's also a language affair. We also know that specialization implies our own vocabulary. It is sometimes need to break walls, but in the end, cross-fertilization is very helpful for creating new dynamics and hopefully revolutionary ideas. This was also an initiative which starts with the idea of looking for local specific problems related to Açores, problems that researchers also have when they are doing research here in this place in Açores. In the end, what we get is a set of proposals which are divided here in the morning in two moments. A moment related more to biology, and the other moment, the next moment, related to technology and education. With respect to technology and education, I will say something about it, and then Pedro will talk about biology. We had some goals in mind, and the challenge was related to education and specifically to mathematics in particular. We were looking for solutions for problems that we have here, and I suppose these problems exist in other places, in other countries. That is how students can learn more efficiently mathematics. And the idea was to try to get some experience from researchers that using different kind of environments related to technology, e-learning, web-based, and to understand how they can be used to promote a better education and to promote a better learning in mathematics. So what we get was free talks. We'll have 3 talks here. The first one will be the path to math, that will be presented by Patricia Jacques, and it is an ETS for algebraic problem solving. Then we have a presentation from Rosa Vicari, Heraclito, a dialectic tutor for logic. And finally, we have a presentation from Elder Coelho with the name A Philosophical View on Artificial Intelligence. Pedro, please.
Okay. Regarding the biology part, why biology? Basically because, in case you didn't notice, this is the Agricultural Sciences Department of the University of Açores. It lives here, so nothing related, at least directly, with informatics, and only indirectly related with biology. But biology is the large field that also contains agricultural sciences in at least part of them. And the other reason is because I'm a biologist. I'm not a computer scientist, although I did start a computer science BSc at the Universidade Aberta, but then I got a permanent position in Finland, and I emigrated, and have given up classes, but not computer science or artificial intelligence. And in fact, if you look at the program, well, the 5 talks in biology have my name, and 3 of them will be presented by me because, well, I guess I was so deeply involved in these problems, in the connection between, especially ecology and artificial intelligence, that I had a number of questions I would like to propose, and in fact I have dozens more ideas that could be put forward, but that will be too much, I guess. And then we tried also to invite some researchers here to also present their own problems, so that you, as computer scientists, specialists in artificial intelligence, could well, say whatever you want. Maybe you have the perfect solution for some of the problems that will be presented. Maybe some will be intractable, at least for now, but the problems will be presented. And the ultimate goal, well, first is to have a discussion on these ideas. Maybe there are, maybe you already have are working in the area. Maybe you right away have some possible solution. So the first goal is to have a discussion on these matters. The second goal is who knows, to have future papers and projects to be submitted to FCT or wherever, if some of the teams are found to be particularly interesting. In the afternoon, if I may say something about it already, we will have a very invited talks by some experts here from the region, and that will talk about very particular problems that the Azores and islands may have, and that may be solved by artificial intelligence methods. So we'll talk about a bit about ecology, about also related with ecology, but in a more applied context, commercial fish stocks, and also geology, volcanoes. I think the third talk is not going to happen, right? So we'll also be around biology and geology. But okay, I think we can
explain the discussion how
what will happen is that, because we have two different teams, first biology and then education, we'll have 3 talks right now until the coffee break at 10. From 10:30 until 1 we have the second part of the challenges, but we'll have a discussion just for the biology questions at 11, after the 4th and 5th talks of the biology section. Then we'll have the 3 education talks and the discussion regarding those education talks. we decided to divide this because there were clearly two very distinct areas that could be related, but in this case there were no connections, so we decided to separate the two things and to have separate discussions. Anything else?
Just say when we start the discussion, we ask for the people who made the talk to sit here and to answer to the possible questions the audience make. Okay, so let's start.
Okay. The first talk will be, is this working? Yes. Will be about automation of species identification and description. Basically, this talk will be about taxonomy. Taxonomy in the context of biology. And what is taxonomy? Basic is naming species, as the name says, but the more precise definition, let's say, it's the science of classifying and describing groups of living organisms based on shared characteristics and giving names to those groups. So I work with spiders, so most of my examples will be about spiders. And so, for example, here is an example of a species of spider. This is an Eresus, Eresidae, from mainland Portugal, another species, and then here another species, and this is the female, this is the male, completely different, as you can see. This is a female, this is a male, so we have here 3 species of Eresus. And then we have a species D in this case, a wolf spider or tarantula, also from mainland Portugal. And in fact, these species, although you can see are really, really flashy, these are really easy to spot with these white or red markings. All these are undescribed. Nobody has ever described these, have no names, these species. But some species are closely related, while other species are very far away in evolutionary terms. So we also group species into other groups. So because these are all Eresus, they are all look-alike, they have a common ancestor relatively recent, so they are grouped into a genus, and this species is completely different, so it belongs to another genus. And so we basically classify species in a hierarchical ranking where species are grouped into genera, families, orders, classes, phyla, kingdoms, and then domains. Right now, we can talk about a serious Linnean shortfall. If you remember, Linnaeus, or Carl Linné, was the first scientist to give names to species as we do it now, with Latin names, so that the names are universal. So we are talking about the same species here, in Finland or in Australia. And there are about 1.5 to 2 million species described, but some estimates say that worldwide we may have between 3 and 30 million species. So all this gap between the, let's say, 2 and whatever, but probably around 10 million species, well, we have 8 million species to be found worldwide. So 80% of the species are not known, have never been seen. We have no idea what they do in the systems, nothing. This is really, really a serious problem. In Portugal, we have about 30,000 species, but known, but maybe we have about 100,000. So we still have to know most of our diversity. This is because very basic science, including taxonomy, is underfunded. It's basically impossible to get money just to describe species. We have to have more other questions, and taxonomy, naming species, describing species, ends up being also, always a side project, a section of larger projects. Even when species are collected, they remain unidentified or undescribed for decades or centuries. These, for example, these are 3 different species, again, of spiders. This is a very particular genus, and all these species, and in fact there are 6 species of this genus, very all look alike, but with subtle differences. And all these species have been captured in Desertas Islands, close to Madeira, where we have 6 new species to science just of this genus that are there, have evolved isolated during millions of years. And in fact, they have been there always, but nobody ever studied this. And the same case for the other red and white species we have seen. Of course, this is information that is lost, and many species may even go extinct before being described. That's, for example, the case of a species of this same genus, of these spiders, that occurred here in Pico Island, that has been found from collections in museums from the 1950s, but has not been found since then. We are pretty sure it's extinct now, and it still doesn't have a name. We just found a few specimens in old jars in a museum. So, okay, but there are 2 different problems. First, it's the identification of described species, because it's pretty hard. As you can see, if you look at those spiders, probably for most of you, they look all alike. Even for me, they look alike. And the second problem is describing the undescribed species, those that we don't even know that exist yet. So the first problem, automatic identification. There are vast numbers of specimens from common species, and this takes lots of time from researchers. Some samples even in very limited sites produce thousands of specimens, and to have a person just identifying all that, it's really time consuming, and we're talking about experts doing this, because if you're not at least trained during 1-2 years, it's basically impossible to tell the species apart. So this problem of automatic identification is an ideal problem for automated processes after having an image library. This, for example, is how we tell. These are all gnaphosids, a particular kind of spiders, and we tell the species apart, in this case the females, by the female genitalia. Basically, all females have different shapes, and then the males have corresponding matching shapes in other parts of the body. This is the underside of the abdomen. So it is possible, it should be possible, to tell species apart if we have good pictures of these particular characters. But truth is, no technology exists to do this. There are a few attempts with very limited taxa. For example, for spiders, there have been attempts with like 15 species from a single genus. There are attempts for some wasps also that can be identified by the wing patterns. If you never noticed, a wasp has a few drawings, let's say, in the wings that are pretty much unique to each species. But this is always in very limited number of taxa. But it could be useful for inventorying well-known faunas, where we already have an image library, and to monitor certain species, for example, pests, which are a pest we have millions and millions of specimens to take pictures of, so that should be rather easy. Of course, there are difficulties, because the interspecific morphological variation may be large, so the same species, the same structures may have some variation. Between species, they may look alike. These, for example, are two similar species, and yet the shapes are pretty much the same of this structure. So the variation between species may be subtle and not really easy to tell apart. The number of potential species is enormous. If we are talking about 30 million species worldwide, or let's say even in a single plot of tropical forest, for example, a single 1 hectare may have 100,000 species. And to know how to tell all these species apart, it's basically an enormous challenge. And then all the problems of the variation of object position and scale, because the specimens may be not really as we like them to be. They may be a bit sideways, or maybe the light is not always the same. So there are a number of challenges here. Some solutions have been tested. The first tries were used traditional statistical methods like PCA, but never really worked. And then now the new way of doing this is with deep networks, and there's been a resurgence of interest with some well succeeded examples, although always with limited number of species. And all of these always require a very rigorous image acquisition. The noise must be controlled, the lighting must be always the same, the scale should be the same, and other parameters. So it's still lots of work for the researcher, or in this case for a technician. The other problem is description of what we don't know. So basically we're talking about automated character extraction. Taxonomists are still using the same methods as 2 centuries ago, as Linné used, although we're talking, in this case, we're talking about morphology. Nowadays we have DNA, we have other techniques that didn't exist at the time, but the morphology is still the same. We look at the microscope and we measure things and we draw things. So recently, some taxonomists have proposed an automated system. They have devised this scheme where you have a robot manipulating the specimen, in this case a wasp, and then you capture images. It goes to a database of images. You separate the different parts, so you have, in fact, a different field for wings, for abdomen, for legs, for head, antenna, etc., etc. And then you extract information from that, and you put all the information back to the database. But this is only conceptual. This has never been done. This was proposed 3 years ago, and nobody advanced in any way with this. So, as you can see, a good system requires robotics to manipulate the specimens, computer vision so that you can see the images, so that the computer is able to take good images of the specimens, and databases that can handle with all this. So the main questions that we thought of are: what are the best existing technical solutions for species identification in these cases of unstructured viewing scenarios? The angle may change, the magnification, etc. What kind of principles should guide the implementation of the image databases? And how to obtain automated character extraction from preserved specimens so that it's easier to describe species and not really relying 100% in a person to extract all the characters. And what kind of characters could or should be extracted? Pictures, and then, but how can we measure, and what kind of structures can we measure? And that's basically it. I hope it was understandable. And well, here are the questions for you to think about, and we'll discuss it later. Okay, thank you. We have now the second talk by Paulo Monjardino, a professor here at the department, about automation of organelle identification, developing maize endosperm transfer cells.
Okay. Hello, good morning. Hope you enjoy the good weather. Could be the other way. Okay. I'm going to talk in a very different structure because I'm going to talk at the cellular level. And this is an ongoing project that has been stalled for the last 2 years because other things had to move on. And now we are getting back to this. I work with maize, with a maize kernel. And this is the image of a maize kernel. Not fully matured, but almost. And this maize kernel has an embryo and has a storage tissue called the endosperm, which makes approximately 75% of it. And in this endosperm tissue, there are a specific number of cells that are very important, are the transfer cells. These transfer cells are in many ways responsible for the kernel yield. And let's put it this way, maize is the most important crop worldwide, the most production, the highest production, and is one of those which productivity has been increasing further and further. These particular cells, the transfer cells, they have a specific characteristic, which is they are specialized for nutrient and assimilate transport. These cells have a very particular features, which are their ingrowths, which is the cell wall folds, and that enables the outer layer, or excuse me, the cell membrane to have a much larger surface than most of the cells have. This image, this image was obtained by scanning electron microscopy, and it was obtained by a group in Australia. This image was obtained by a confocal microscope, and it was labeled for cellulose only, and it was obtained here in my lab. These are different technologies, but they give you an interesting view of how these cells can have these specific features of the ingrowths that I told you about. Most of the ingrowths are linear longitudinal, which they call the flange ingrowths. In our case, we reported the existence of reticulate ingrowths, which we published this year. It's something that everybody said it did not exist, but it was not too hard to demonstrate. It was the other way around. In any case, the most important issue is these particular cells are very determined for flow of assimilates into the endosperm and therefore for production. And there are some patents regarding these cells at this present stage. They are important for assimilate flow because actually they promote two types of transport. Passive transport, which is mediated by enzymes. Actually, they are mostly three. Or for passive transport or for active transport associated with something that some of you know what it is, which are the proton pumps and specific transporters that work with Symport for hydrogen ions and sucrose. Sucrose is the most important assimilate that flows into it, but it's not the only one. Amino acids are also very important. Well, this image will move on because I was hoping we could see it here, but we cannot. Anyway, these cells, they have the developmental features. They start developing these ingrowths at very early stages, and as they mature, these ingrowths become more pronounced, up to 6 layers inwards. And what we are aiming at is how these ingrowths are formed. If we want these cells to be more efficient, we can have more cells with ingrowths, or we can have more developed ingrowths in order to make them more efficient for assimilate flow. And if they are more efficient to assimilate flow, instead of having kernels that weigh around 0.25 grams, we might have eventually kernels that weigh about 0.35 grams. If you multiply this by 600 to 800 kernels per year. And if you multiply that by 75,000 years per hectare, you might come to an interesting number of significant increase in yield. As I was an undergraduate student, we would consider a good yield of maize as 10,000 kilos per hectare. 10,000 tons, that's too much. Nowadays, we consider a good yield about 16,000 kilos per hectare nowadays. And we believe that we will get to 20,000 kilos per hectare in the next, 5 to 10 days. At least the demand for this crop are this high, and this, I believe, is a realistic estimative. And to manipulate these cells is one way of getting there. Okay, so what did we do in order to understand? This is the reason why I came here. What we do in order to understand how these cells develop? We use the transmission electron microscopy in order to assess how they develop and obtain images, a magnification of 12,000 times. 12,000 times for a transmission electron microscopy is not too much, but it's good enough for us to identify every specific organelle and structure within the cell. If we do it higher magnification, we have even bigger files, and it's not very practical, as you will see. And we did it on a developmental scale, which is from early initiation stages up to fully matured cells. We overlap a grid of 1 by 1 micron on top of the photomontages obtained by this microscope. And we introduced the data of the intersections of the grid into an Excel file. Pedro, which is one of the co-authors of the presentation, developed a small program, a short one. It was very quick, actually. And basically, what we did was to calculate the shortest distance of each specific organelle to a specific reference point. Reference point can be another organelle. Reference point can be a cell wall. Actually, we basically use the walls. Oh, excuse me. Wrong button. Okay. We use the walls, the ingrowths, and the nuclei as reference points. Because biologically were the ones who made more sense to us, but actually could do to anything else, as long as we were able to label it. Okay. So just to give you an idea, this is a cell, this is a group of cells at very early developmental stages. So we overlap a grid 1 by 1 micron, and in each intersection we score what it was there. This is where the automation could be interesting because it takes a lot of time to do that. And the other thing, it can be prone to error. And what we did also afterwards was to outline the walls and outline the nucleus. In this particular case, we could only see one nucleus, and why so? These cells are approximately 20 to 40 by 20 to 40 by 20 to 40 micro size. But each specific section is about 50 nanometer width. Therefore, the odds of finding a nucleus in a specific section is less than 1%. So actually, yes, it's less than 1%. Therefore, we could only see one nucleus in one particular cell, but that's okay. We don't have to reference it always to the nucleus. The ones we were more interested on was to reference actually to this particular cell, wall, which we call the outer periclinal wall. So what was the output of this? The output is an Excel file, which this is all done by hand, usually by two people. One that is identifying, and the other one is scoring, and is debating whether or not something is what we think it is. I'm the person that is identifying, and the person that is next to me is the one that is scoring. And this gives you a glimpse of the letters we used to identify the organelles or structures. And we have one very important, which is a Z for unidentified organelle or particle. And that happens. And that happens quite often, actually. If you're not sure, it happens like this. Usually the files are this big. I'm not asking you to understand what it is, but it gives you a glimpse of what the sizes are. And then we can do analysis specifically with the algorithm that Pedro developed, and then it gives us minimum distances of organelles towards points of reference that we are interested on. The challenges we find in this methodology is that it can be difficult to identify organelles, and I'll give you examples how difficult it might be. Some unknown structures can happen, and those are the ones we score as Z, because we cannot identify. Usually vesicles can be tricky. Peroxisomes are not very common in these particular tissues. Small vacuoles and others. If we have doubts, we score as Z. If it's a Z, it means that we exclude from the analysis. So we try to be as conservative as we can in this regard. The organelles, and it's a bit like Pedro spiders, can have distant looks throughout development, and actually, depending on the quality of the fixation procedures that we use when we process these samples. And the challenge is how repetitive the analysis can be, which is, if I go again in this specific data, will I score things the same way as I did before? I tend to believe so, but it can be tricky sometimes. And of course, the quality of the images can be impaired by the quality of the fixation, and this is not a small issue. And of course, the process can be very painstaking. We deal with very large files, often larger than 1 gigabyte, and of course, this is a time-consuming process. This is all the challenges I could come up for this presentation, and I think these are the most important ones. Just to give you an idea how these things look, this is a cell at 6 days after pollination, which is at the early developmental stages. This is the outer periclinal wall, and this is a unique ingrowth that this particular cell developed. This is mitochondria, this is Golgi apparatus. Some of you might remember that from high school, I guess. We have rough endoplasmic reticulum. It's enlarged due to some fixation problems. This is a vacuole with precipitates inside of it. And you'll see that some of these things shift in their appearance as we go through different samples. This is in a more advanced stage. At 10 days after pollination, the fixation procedure is a bit different from the other one. We evolved because we needed to do something about that. And this particular thing was very surprising for us, because actually these are vesicles that are fusing out to each other. And in a large group of samples, we came up with this whitish kind of thing, which the literature did not reference this before. And we were surprised with that, but the data was consistent. Of course, these are some of the ingrowths they have, and these actually are easy to identify. So this is a bigger magnification of those vesicles that are very uncommon in their looks, because usually the vesicles look like this. But all of these are ingrowths. This is mitochondria, not a bit different from what you see in textbooks. And also in the middle of this, we have mitochondria. For instance, this is a cell wall, goes around like this. These are plasmodesmata, and these are all ingrowths. And to put this in an automatic system, I really don't know how to do it. I hope that some of you may come up with some interesting recommendations. It would be interesting. And this is, for instance, these are, for instance, cells at a more developed stage. And these ones have more developed ingrowths, the two types, the reticulate and the flange ingrowths. And in the middle of these, we can have mitochondria, for instance. And mitochondria is the easy organelle to look at. This is the easy one. It might look like this. These are all at the same stage. Here they are much more clear. Here they are more diffused. Or in this particular one, their cistern, which are these internal membrane systems, are not as clear as in these particular ones. But let's put it this way. These are the best images. Imagine what the worst image look like, okay? And how they can vary. And all of these are ingrowths in the middle of these. It's easy to spot ingrowths because these are fibers. These are cellulose fibers in the middle of these, okay? And of course, what can complicate this a little more is that among these organelles, among these mitochondria and these ingrowths, we can see, I have to use this. It is okay. For instance, there is a vesicle here where, oh, the arrow is gone. Oh boy, go back. Because in the screen I have more difficulty to see this. Not in this screen. There's a vesicle here, and this is mitochondria. This is tricky. And of course, that's why I'm usually the one that does the measurements. Because if I do a mistake, at least it's a systematic mistake. It's always the same type of mistake, because different people might have different interpretation. That's why you usually have two people to make a score like this. And this is the same stage, but at a different part of the cells. And this gives you an idea how tricky this can be, because the cell wall goes like this, goes down here. Some plasmata can be seen, and all of these are ingrowths. How can the computer say, or how can some program say, this is an ingrowth, and this is the cell wall? It's not easy to say. Therefore, we raise the question, of course, that's why we're here, hoping to hear a miracle. Organelle identification is one of the big challenges here. It's not as simple as the textbook. The textbooks often mislead us. It's critical to make a good distinction between wall types and respective ingrowths. If you look at the general picture, it's easy to do it. If we do it on a more detailed image, it might be more difficult. The process of data gathering, if we could get a way of doing it, like for instance, the big Excel file that we have there, it took us like 4 or 5 hours to get that file. And we have to have many of those in order to have repetitiveness, and then we can do the statistical analysis. And of course, other challenges that we have not come across, we are happy to hear from you, as smart people you all are, that can give you some glimpse, some idea of what this could be. So I look forward for the discussion, and thank you for your time.
Okay, it's me again. Okay, this is another problem, which in fact is somewhat related with my first problem. It's about biodiversity data compilation. There are a number of biodiversity data portals that are in fact increasing in number and scope. The main worldwide portal is EOL. Let me see if this works, which is the Encyclopedia of Life, which basically wants to list and describe all the species around the world. So, for example, let me see if this works. Will this identify? No? Okay. Nothing. But anyway, this is let's see, one. This is, of course, under construction, but for each species worldwide, it has pictures, it has a number of data, like some papers that talk about this species. We have distribution. There should be maps for many of these species, of course. Many species don't have any maps at all, or don't have any good information, because they are simply basically unknown, although they have a name. And then we have, at the national level, we have Naturdata, which is a portal, a national biodiversity portal that in fact I'm coordinating together with other persons, of course, and it lists all the Portuguese species, all the known Portuguese species. Ferradura, let's see if this finds, for example, morcego de ferradura. Rhinolophocyposidерus. And then you have pictures, you have the taxonomical classification, you have different data. You have, for example, the weight, the body length, some morphological description. So whatever data the collaborators want to put here. And this is all voluntary, so anyone can contribute. And you have a distribution map, which is, of course, very incomplete, simply because nobody has put data into the portal. Okay, and then the Azorian Biodiversity Portal, which is coordinated by Professor Paulo Borges, which is sitting here among other people, like Professor Rosalina. And this is specific for the Açores. So for each species, you have the same type of information. For example, if it is endemic to the Açores here, so if it's exclusive to these islands, you have a distribution map, etc. And this type of portals is rising, and this created the need for biodiversity information standards, because these portals usually cannot communicate. So this TDWG is a standard that says that each portal should have, in order to intercommunicate with the others, a given number of fields with given names, with given characteristics. But whatever portal we're talking about, this requires huge resources. You can imagine listing information for the hundreds of thousands of species. In the case of EOL, for millions of species, it's a nightmare. This is usually done with voluntary contributions. In the case of Naturdata, it's 100% voluntary. We never had any financing for this. And the fact is that many professional and amateur naturalists do gather valuable data in a number of places. Databases, their own databases, different databases, websites, social networks. Of course, there are the typical papers, research articles, a number of documents spread all over the internet. But these don't communicate, so none are easily searched or retrievable. Of course, we can talk about a semantic web, where everything is data, not text, so that the computers know what is what, so that they can compile the information to a single page, but this still has to come. So right now, we need ways of retrieving and compiling data from these disparate sources. Maybe a model is Wolfram Alpha. I guess most of you know Wolfram Alpha basically is a portal that tries to compile information from different sources into a single portal. So for example, if you write here Azores, you see what it gives. But basically this portal, from what I know, is gathering information from different sources and is trying to find out what is what, and is compiling all the information in a single page and creating links, etc. And none of this is really here. It's not known by the portal, but it's being created on the fly from different sources. So maybe this is a possible model. This kind of portal. Of course, for species, for biodiversity, some data are easier to find and to digest. For example, the common names. Here you have the example of common names of butio-butio, which in Portuguese is Águia de Asa Redonda, in English buzzard, here in the Açores milhafre, in Madeira manta, etc., etc. So these have different names according to the region, to the country. Sometimes in each country they have even different names. Maybe it's easy to get data, for example, from the size, maybe from the trophic gills, if it's a predator, if it's an herbivore, from the habitat they live in, here the Laurisilva, which is a typical forest here in the islands, maybe they live in caves, in deserts, maybe data from the distribution, which may be in a number of different of different, maybe presented in a number of different ways, maybe coordinates, maybe the name of the site, maybe just the country. Maybe it's easy to extract threat status, like red lists, if national red lists, international red lists. Maybe it should extract information from legislation. Maybe you could go and get pictures and videos and try to find out if those pictures do match the species. So the questions here are, is it possible to create such a search and ingest engine for a biodiversity portal? And then what kind of information is easy to collect and which is difficult to deal with, and we should not even bother about it. Because we are a bit late, but I think I recovered the time. I'll leave with these two questions. And although we have the discussion later, of course, we have now the coffee break during half an hour. I'll be here, and Paulo, I think, will be here. So if you want to ask anything before the general discussion, be free to do it. Okay, thank you. Was a coffee break? No?